Good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Current Status of Surrogates for Survival in Cancer Trials. My name is Vicky, and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, with time for a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. And if you do require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in a listen-only mode. Please note that this event will be recorded and made available for future download. At this point, I would like to thank IDDI, who helped develop the content for this presentation. The International Drug Development Institute is an expert center in biostatistical and integrated e-clinical services for pharmaceutical and biotechnology companies in several disease areas, including oncology and ophthalmology. IDDI optimizes the clinical development of drugs, biologics, and devices thanks to proven statistical expertise and operational excellence. Founded in 1991, IDDI has offices in Belgium, Boston, Raleigh, and San Francisco. And now I would like to introduce our speakers for today's event, Dr. Mark Boyes and Dr. Everardo Saad. Mark holds a SCD in biostatistics from the Harvard School of Public Health. He is the founder of the International Drug Development Institute and of CluePoint, two biostatistical service organizations based in the US and Europe. He is interested in clinical trial design, meta-analysis, validation of biomarkers and surrogate endpoints, statistical methods in oncology, statistical detection of errors and fraud, statistical monitoring of clinical trials, and medical data sharing. And our next speaker for today is Dr. Everardo Saad. Dr. Saad is IDDI's senior medical expert and has over 15 years of experience in medical oncology and clinical trial designs. He graduated in medicine and trained in internal medicine in Sao Paulo, and did his fellowship in medical oncology at the University of Texas MD Anderson Cancer Center in Houston. After practicing for several years, he shifted his professional career towards education and research in medical oncology, and has a special interest in clinical trial methodology, the assessment of endpoints, and the development of novel therapies for cancer patients. And now, without further ado, I would like to hand the mic over to our speakers. You may begin when ready.
Thank you, Vicky. Hello to all of you. Thank you very much for attending this webinar. We hope you will enjoy it. So today, Mark and I will cover the following key issues. First, I will make an introduction and we'll discuss some of the pros and cons of some of the most important efficacy endpoints for clinical trials. Then Mark will come and talk about the preferred method for validation of surrogate endpoints with various technical details about how this is done and how this is interpreted. Then I will briefly discuss some of the results that have been published so far, especially for disease-free survival or DFS and progression-free survival or PFS as surrogates for overall survival. And in the last few slides, I will briefly discuss the evolution of surrogacy within a given setting and also potential differences between treatment classes for the same disease type. Now, let me first start talking about therapeutic objectives. In essence, this translates into questions such as what do patients expect from treatment and what do we physicians want to accomplish with treatment. And of course the answer is number one here. It's cure. So this is this would be the ideal endpoint in any clinical trial and the ultimate therapeutic objective in clinical practice. However, since this is not possible in many situations in advanced disease, extension of survival is probably what's most important, although maintenance or improvement in quality of life is also relevant. So I looked for the current evidence for these objectives, and there is some research published, and it seems, and some of it is summarized in this very nice paper in the Journal of Clinical Oncology. So it seems that patients' perspectives are often different from those of the well person and also of healthcare professionals, like physicians and nurses and other types of professionals. And there are many surveys, and when patients are asked explicitly, they tend to accept toxicity in exchange for relatively small health benefits, especially in survival. And in most surveys, it appears that patients tend to give more importance to survival than well people and healthcare professionals. Conversely, well people and healthcare professionals, perhaps because they don't have cancer at the moment, seem to be more concerned with quality of life than with survival. Whereas in some surveys, the reverse seems to be true for patients. So both objectives are important, survival and quality of life, but it seems that for patients, survival comes first. Now, the question, what is survival, may seem like trivial or hair-splitting about a term, but I think it's very important because my impression is that there is some confusion in the role that survival plays in different situations. For example, at least in 4 different dimensions, survival can be seen as an outcome desired by patient. It can be seen or thought of as a therapeutic objective to be pursued by physicians and healthcare professionals. It is, of course, a clinical trial endpoint, as we will discuss for most of this webinar, and it's also a public health measure because we have to think about the effectiveness of interventions in addition to their efficacy. So the goal here is to make the distinction between survival as a therapeutic objective and as a clinical trial endpoint, and this could be conceptualized in different ways. And one of the ways is these questions here. What do we want from these in these 4 dimensions? So the questions we ask when we think about a therapeutic objective, something to be achieved in our patients in clinical practice, are questions such as the following: can survival of groups of individuals be prolonged? If so, by how much? For example, by thinking about difference in medians or averages, if they are available. And also we think about, are there treatment characteristics, patient characteristics that affect the treatment effect? Does it vary across patient types? Now, when we think about survival as a clinical trial endpoint, our main concern is whether can any eventual impact of the treatment on survival be demonstrated, statistically speaking, with reasonably small sample sizes and trial durations. In other words, are the resources available? Do they allow for this demonstration? So when we think about a clinical trial, we think about the metric that we have to use to capture the differences in survival. And it's not the same kind of question that we have in patients. We want to extend their survival, of course, but we have nothing to compare with. So these differences are relatively subtle, but I think they affect the way people interpret the role of survival as an endpoint. And I hope to make that clear in the next few slides. There is some confusion out there. For example, even in this very important FDA guidance on clinical trial endpoints for oncology, the notions of endpoints and the notion of clinical benefit in a way they get mixed up. So the document says that for later phase efficacy studies, in other words, for phase 3 trials, an endpoint should assess whether a drug provides clinical benefit. And then clinical benefit is defined in this document as either important clinical outcomes, such as improved survival or improvement in symptoms, or effects on established surrogate endpoints, or still effect on a surrogate endpoint that is reasonably likely to predict clinical benefit. But in my mind, these things are very different. And when we think about increased survival or symptomatic improvement, in other words, clinical outcomes, I think we're best left with the notion of clinical benefit, which to me equates with therapeutic objectives, not with clinical trial endpoints. Endpoints, the way I see it, are metrics that we use to represent the efficacy or the safety of treatment in a clinical trial. And I'm going to try to show why. If we think about endpoints, overall survival is no longer as relevant as it used to be, although it remains the ultimate therapeutic objective in clinical practice. So what is the ideal endpoint? It should at least capture all the clinically relevant events. It should be easy to measure. It should provide little opportunity for ascertainment bias. It should be observed as early as possible and is in as many patients as possible. And of course, it should be statistically sensitive. It should be easily, the differences in this endpoint should be demonstrated with reasonable numbers of patients and reasonable power. So does the ideal endpoint exist? And here we try to show, at least with regard to these 5 features here, whether something is good, not so good, or really bad. So if we think about survival, quality of life, time to progression, or if you will, PFS, which is not exactly the same, but for the sake of this discussion, we can think of them as the same. Response rate, none of these fulfills many of the requirements that we saw in the previous slide. So none of them has features that allow us to say that they are really good endpoints. Whereas a biomarker, or at least an idealized biomarker, would probably be would be closer to the ideals that we think in terms of these characteristics, although arguably they don't have clinical relevance, because of course measuring a biomarker is not what will change a patient's duration of life or symptoms. But if we can develop biomarkers that are easy to measure, quickly measured, which have very little potential for bias because they're really validated analytically, and with statistical power because they are more sensitive to changes in the disease biology, these would be the ideal endpoints. But of course, they don't exist at this point, and the search continues for such biomarkers. So we have to remain for the time being with the endpoints that we all know, and these are the ones that we are mostly discussing during this webinar. Now, if there is a hierarchy, or at least historically there is a hierarchy of efficacy endpoints, and overall survival, OS, of course sits at the top of this hierarchy. All the others come after it. So survival is seen historically as the most important endpoints. But there are well-known problems with OS, and a number of reasons why OS is more and more difficult to be used as primary endpoint, especially because of the number of events that are needed to detect survival differences. There will be smaller events with the OS than with any other endpoint for obvious reasons. The time it takes for death to occur, of course, is longer than for a response or a progression to occur. There is a smaller relative effect on OS than on other endpoints typically, and then more and more and this is becoming an acute problem in some disease types, there are competing risks of death and crossovers. So I summarize here what I said in the previous slide, showing simulations or actual real examples from the literature. For example, with regard to the smaller number of events, if you think about the number of patients represented here over time, the number of patients accrued in a study, not all of them will have the events of interest. And if we think about, for example, PFS events, in other words, progression or death, there will be a number of patients having these events with a certain distribution, and the number of deaths will always be less just by definition. Then if we think about longer median times to event and small relative effects, if we compare the PFS as an endpoint and know that, for example, a certain treatment improves PFS by leading to a 4 months difference in medians, if this difference in medians is carried over to OS, since OS is much longer than PFS on the time axis here, the number of events that it takes to find statistically significant differences in PFS is much smaller than the number of events to find differences in OS. And the relative benefit of treatment is also different, as you can tell from a hazard ratio of 0.75 for PFS and 0.86 for OS in these two simulation examples. Now, in reality, one problem that is particularly of concern nowadays is the increasing role of subsequent line therapy in determining patient survival, and this can come in the form of crossovers in studies. But there is also situations where there are competing risks of death, which may eliminate potential survival differences and make treatments more similar when we analyze survival. For example, for crizotinib, which is very effective in non-small cell lung cancer with a particular type of mutation, the progression-free survival is significantly improved by the use of crizotinib, and this is, for example, illustrated by a very low hazard ratio of 0.45. But when survival is analyzed, this situation is now being repeatedly observed. There is no difference between arms, essentially because patients that are treated with the control treatment, in this case chemotherapy, they are at some point either as foreseen by the trial or in clinical practice, they can cross over to the experimental arm, and then their survival tends to be not different from a statistical point of view. So this is, to me, the most pressing issue of why OS is less and less a relevant endpoint, or at least a less relevant primary endpoint. It should always be a secondary endpoint. So this sets the stage for surrogates, and surrogates are endpoints that can replace so-called true endpoints, usually because they are assessed earlier or using fewer resources. But of course, based on what I've just discussed, the question arises, are there true endpoints? And of course, that can be discussed, but if the answer is yes, despite the limitations, there are still OS and quality of life. Now, quality of life, as many of you will know, is not often used as primary endpoints in oncology, and there is much less convincing evidence in the literature of using quality of life as an endpoint to really decide between competing treatments. So we're left with OS as the primary endpoint of most interest as a true endpoint, and that's in this background that we will further discuss surrogacy. Now, why are surrogate endpoints needed? Mostly they're needed to reduce the development time. If we use surrogates that are observed earlier, and we know that they predict for the treatment effect on survival, they can expedite drug development. But also because if the surrogates are more commonly observed, for example, I mentioned that PFS. events are more frequent than OS events, this will increase the statistical power and therefore will be useful for drug development as well. So at this point, I will hand the presentation over to Mark, and he will discuss more technical issues about surrogate validation.
Thank you, Everardo. Now I would like to go into the statistical part of this webinar, where we will look at some statistical approaches that have been proposed to validate a surrogate endpoint. So as Everardo made clear, a good surrogate is one that can substitute for a clinical endpoint. And this is really the topic that I will expand on in the next few slides. It's important to note ahead of the next few slides that a simple correlation between a surrogate endpoint and the true clinical endpoint is not sufficient to make it a good surrogate. And I will come back to this notion repeatedly, that this is really a key aspect of our discussion here. Because what we want, in addition to correlation, we want a surrogate endpoint to be able to predict the treatment effect. So, and the treatment effect can be, of course, a benefit, a harm, or a lack of both benefit and harm on the clinical endpoint. So we want to be able to predict an effect. And this is absolutely key in the discussion of the statistical methods that have been proposed for validation. The earlier attempts of statistical validation were due to Ross Prentice back 25 years ago. And Ross Prentice proposed essentially 3 criteria for surrogacy, which are listed here. In order for a surrogate to be valid for a clinical endpoint, the surrogate must have an effect on that clinical endpoint. The treatment must have an effect on the clinical endpoint. And third, and most importantly, the treatment has no longer an effect on the true clinical endpoint if we adjust for the surrogate. So I will describe this in a graphical manner on the next slide, but before I go to the next slide, we need to note that, first of all, condition 2 here, or criterion 2, which is that the treatment should have an effect on the true endpoint, is not always fulfilled. If the treatment were known to have an effect on the true endpoint, then by definition we would no longer need a surrogate. So in a sense, it's very hard to argue that you need to show an effect on the true endpoint in order for a surrogate to be valid, simply because if that were the case, you no longer would need it. You no longer would need the surrogate. And so that's kind of a conceptual problem. The second conceptual problem with Ross Prentice's criterion is criterion number 3, really raises major conceptual problems. And I will not go into these today, for the benefit of time, but in fact, condition number 3 has been shown to be quite, not only quite difficult to fulfill in practice, but even to have some mathematical drawbacks that make it really difficult to use. Now, let's try to explain the criteria from Prentice in a graphical manner. We really have 3 kind of entities to look at: the randomized treatment in a clinical trial, the potential surrogate that we have interested in, and the true endpoint. And so what we are interested in first is that the surrogate must have an effect on the true endpoint. This is one of Prentice's criteria. The second criteria is that the treatment must have an effect on the true endpoint, on the clinical endpoint. And the third criterion is that if we adjust the effect of treatment on the true endpoint for the surrogate, then there is no remaining effect. In other words, the whole treatment effect on the true endpoint is in a sense captured by the surrogate. And so that is, like I said, the condition that is not just difficult to fulfill in practice, but mathematically very challenging to show. It can be shown, by the way, but it requires use of causal inference methods, and there hasn't been a lot of them published yet, but some people are working on these issues at the moment, and the methodology required to show this third criterion is actually quite challenging. Some colleagues and I have proposed a different approach to surrogacy, which has become quite popular in recent years. And the 2 conditions that we proposed to be fulfilled for a good surrogate are listed here. The first one is identical to, in fact, one of the Prentice criteria, which is that the surrogate must have an effect on the true endpoint. So this is what we call the individual level surrogacy. It's the surrogacy at the level of the patient. For a patient, suppose the patient has longer PFS you expect that patient to also have longer OS. Conversely, if a patient has a very short PFS, you expect that patient individually to have a shorter OS. So that kind of makes sense intuitively that the two endpoints, the true and the surrogate, must have some correlation. Otherwise, you would really doubt that the surrogate has biological meaning. The second condition we propose, which is very different from Prentice's criteria, is that the treatment effect on the true endpoint can be predicted from the treatment effect on the surrogate biomarker. So we really use this notion of being able to predict an effect. And so how does this work in our graphical presentation? Just like in the Prentice case, we want to show that the surrogate and the true endpoint are correlated. So in other words, the surrogate has an impact on the true endpoint. And secondly, that the effects of treatment on the surrogate and the true endpoint must be correlated. So we look at the effects of treatment on the surrogate and the effects of treatment on the true endpoint, and we must have a correlation between these effects. Now, the first condition, that the surrogate and the true endpoint are correlated, can be shown easily in a single trial. It just consists of showing that the surrogate is a prognostic marker for the true endpoint. So that's very easy to do. You don't need that many observations. So that's really a piece of cake. What's difficult is to show that there is a correlation between the treatment effects, because this, by definition, requires several trials. Unless you look at causal inference approaches, where you look at the causal effects of treatment, which we will not go into here today. If you want to look at the treatment effects on the surrogate repeatedly, then by definition you need, a meta-analysis of several trials in order to have repeated observations of the treatment effect on the surrogate and on the true endpoint. And then you want to show that these are correlated. So let me show you an example, and I hope this will make this concept really clear. Patricia Cortazar at the FDA, the Food and Drug Administration, and colleagues carried out a surrogate validation using this approach. And they were interested in a situation which is shown here, situation of neoadjuvant chemotherapies for early breast cancer. So breast cancer patients who could receive chemotherapy prior to surgery in a neoadjuvant setting. And then the OS, overall survival, was the true endpoint of interest, and the pathological complete response, which was the response seen at the time of surgery, was the potential surrogate here. So using the meta-analytic approach to show that PCR can be used as a surrogate for OS in early breast cancer requires two conditions to be shown. First, there is an association between PCR and, it should say OS here. They looked at both OS and EFS, event-free survival, but I'm going to concentrate here on OS. And then secondly, they need to show that in several trials, there is a correlation between the effects of treatment on PCR, and that is measured by the PCR odds ratio, and the effect of treatment on OS. Sorry again for the typo, this should be OS, not EFS. And this is measured by the hazard ratio of these treatments on OS. So here are the data that they used to do this. They had a large database of about 12,000 patients, and they obviously had individual patient data for all these patients. These patients had resectable breast cancer, and they had been randomized in 12 different trials to various preoperative chemotherapies. The details of the treatment comparisons don't matter, but each trial provided one or more comparison between randomized treatment groups. So the treatments were, again, we don't go into what treatments exactly they looked at, but in every trial they had defined experimental groups and the control arm. So they could compare the experimental arms with the control. The surrogate, as I said, was PCR, and the true endpoint, or the clinical endpoint of interest, was OS. First condition, again, is to show an association between PCR and OS. In other words, show that the PCR, pathological complete response, has a prognostic impact on overall survival. And this is shown on this slide here. What you clearly see is that patients who did enjoy a PCR, the curve in red here, had a much better survival curve, a much higher survival curve estimate than patients who did not reach a PCR. So patients who have a PCR have a better survival, and this is captured by the survival hazard ratio here of 0.36 in favor of patients who had a PCR versus patients who didn't. So this is a major, obviously a major prognostic impact of PCR. Again, is that sufficient to claim surrogacy? it is not. Because you need to look also at the association between the treatment effects on PCR and on OS. They had 12 trials, so we have 12 points here. Each point represents one of the randomized comparisons of interest, one of the trials, if you like. And what this graph shows is the association between the odds ratio for pathological complete response, the x-axis, versus the hazard ratio for overall survival, the y-axis. So this, in other words, looks at the association between treatment effects on the surrogate and the true endpoint in these 12 trials. And what you clearly show here, even without doing any sorts of fancy calculations, is that there is very little association, indeed, between these treatment effects. What we call the R squared for trial, which is a statistic of association between the treatment effects. It's called the coefficient of determination of a regression line here that you could fit throughout these points. The R squared is only 0.03, which is, in other words, reflecting the fact that the difference between the treatment arms in terms of odds ratio of pathological complete response explains only 3%, 0.03, of the variability in the treatment effects on overall survival in the same trials. So if you see an effect on complete response, pathological complete response in a trial, that does not allow you to claim that there will automatically be an effect on overall survival. So we see a very poor correlation here between the treatment effects. And we could ask the question, what would have we expected for a good surrogate? Well, for a good surrogate, first of all, we would have expected, rather, that the lack of effect of the treatment on the surrogate, an odds ratio of equal to 1 for PCR, would actually predict no effect on survival. So and indeed, if treatment does not influence the surrogate, then you would expect that treatment does not influence the true endpoint for a good surrogate. And so this point here should be part of our regression line. And then you would also expect that for a trial like the NOAA trial, which actually I didn't see before because this was a very small point. The NOAA trial didn't have a lot of events, but if you look at this point here, shows that the NOAA trial, which was one of the 12 trials in the meta-analysis, they had an odds ratio of 3. So the effect of treatment in the NOAA trial was major on pathological complete response, and it was also a major effect on overall survival, since in the NOAA trial, the hazard ratio in favor of treatment was 0.6. So indeed, the NOAA trial showed a large effect on both the surrogate and the true endpoint, and if you now assume that no effect on the surrogate and the true endpoint should also be part of the regression line, you can in fact draw this kind of idealized regression line, the one that you would have expected for a good surrogate. And it's obvious that the observed line, which is the horizontal line here, is very different from the expected line for a good surrogate, which is, in other words, saying PCR is just not a good surrogate for OS, at least in this particular set of trials. And in fact, people were very bothered by this. Everybody expected PCR to be a good surrogate for OS for many reasons, because it makes biological sense, and there is every reason to believe that if you can induce a response in some patients, then they will also live longer. And that doesn't seem to be the case, again, in this limited set of 12 trials. So people tried to explain, and there is a very thoughtful discussion in the Cortazar paper on why this was a lack of association instead of the expected association. The patients were very heterogeneous. There were different, breast tumor type. There was treatment heterogeneity. All of these trials looked at different types of treatments. There were some that looked at anti-HER2 therapies, others that looked at chemotherapy. So that was a mixed bag of different types of treatments. There were many other sources of heterogeneity, and last but not least, the treatment effects on survival and even on PCR were pretty small, actually, except in the NOAA trial. The NOAA trial had a large effect, but many other trials had a really small effect on PCR and also a smallish effect on survival. So, I mean, this could explain why we don't get a good relationship here. In spite of this, it's interesting to note that the FDA had written a guidance for industry to the effect that pathological complete response can be used as an endpoint to support accelerated approval. And interestingly, despite the kind of negative result they found in the meta-analysis, that guidance is still current. So PCR is still accepted as an endpoint for accelerated approval, even though we cannot claim, at least statistically, that it is a good surrogate for survival. So let me turn to some other situations, and I would like to take one in particular, which is the situation of gastric cancer. And the reason I like this example is that we have done two meta-analyses, one in which we looked at early patients who could be resected, in whom the tumor could be resected, and in that set of trials we looked at disease-free survival as a surrogate for overall survival. And then we had a different set of trials in patients with advanced disease, and in those patients we looked at whether PFS, progression-free survival, could be used as a surrogate endpoint for overall survival. And quite frankly, if you think of the problem before you do the calculations, you would think, well, it's likely that PFS is a pretty good surrogate for OS because, you know, death occurs soon after progression, and DFS may not be such a good surrogate for OS because there is a long time between patient, the disease recurrence in a patient and the fact that this patient will eventually die of gastric cancer. So if you had to bet on anything, you would say, well, PFS may be a good surrogate for survival and DFS probably isn't. So we will look at the data, and if the data confirm our prior expectations. So we had two data sets, as I said, one in localized gastric cancer, about 3,000 patients, 14 trials, and we had 5 extra trials that we did not include in the calculations. We used just those for validation. And then we had 20 trials in advanced disease, 4,000 patients, and we had another 12 trials that we kept for validation. So first, and I'm not going to talk about individual level association, I'm going to look at the predictive effect of the surrogate. In other words, whether the treatment effect on the surrogate predicts for the treatment effect on the true endpoint. So I'm going to focus on the trial level association. So first, looking at localized gastric cancer, this is again a curve showing the treatment effect on disease-free survival. It's the hazard ratio versus the treatment effect on overall survival. Again, the hazard ratio for OS. And what we see here is all the trials, again, each point is a trial here, all the trials that we had data for, and it's fairly it's easy to see that there is a very strong association in contrast with the pathological complete response versus OS treatment effects that I showed before. Here we have a really good correlation, you know, this shows that there is a very good trial level association. Now we want to go one step beyond, and we want to look at something we call the surrogate threshold effect. The surrogate threshold effect, which was proposed by my colleague Tomasz Borzykowski and myself, is the treatment effect on the surrogate that predicts for a statistically significant treatment effect on the true endpoint. And this is obviously what everybody wants. To say, well, if we exceed the surrogate threshold effect, then we're very likely to have a significant effect on the true endpoint of interest. And that is, of course, in practice quite important. Now, it turns out, if you look at the prediction limits around the regression line here, that the surrogate threshold effect is the point at which the upper prediction limit, in fact, intersects the null hypothesis on overall survival. So the intersection between the null line here and the prediction limit, the upper prediction limit of the prediction line, in fact, that point is the surrogate threshold effect. It turns out to be 0.92 here. So what does that mean? That means if we did a trial and if we observed a hazard ratio of less than 0.92 on disease-free survival, we could say, based on this data, well, we're pretty sure, not sure to 100%, of course, because statisticians never are sure to 100%, but we're pretty sure, 95% sure, in fact, that there will also be an effect on overall survival. So does that work? Well, you know, we had validation trials, and indeed, when we looked at the 5 validation trials that were not part of the trials that we used for the prediction we just showed, if we look at these 5 trials, 3 of them had an observed effect on survival. See here, the 3 trials in red, they had a significant effect on overall survival, and 2 trials didn't. And lo and behold, if we look at the prediction simply based on the hazard ratio on DFS, which is here, simply based on that hazard ratio and on our prediction line, we see that we would have predicted a hazard ratio on survival that reaches significance in all 3 cases. And in the two cases that, in the two trials that were not significant for OS, we would not have predicted a benefit on OS. So perfect prediction. Of course, it's only 5 trials, but perfect prediction of the 3 that actually reached significance. And it's shown here. You see the 3 trials for which we predicted a benefit on OS, in fact, had a big effect of treatment on DFS, and that's why we predicted a significant benefit on OS. And these trials here did not have a large effect on DFS, therefore we would not have predicted a large effect on survival. So DFS is a pretty good surrogate for OS, perfectly predictive in this set of trials. In contrast, when we now look at advanced disease, advanced gastric cancer, PFS and OS, the effect of treatment rather on PFS and OS, are much less well correlated. You see that regression line here is much less convincing than what we had before, and the prediction limits are much, much wider, which then implies that the surrogate threshold effect will be much smaller. In other words, the PFS that predicts for an OS benefit is a much smaller PFS hazard ratio. And so here, the trial level R squared, the coefficient of determination is 61%, which is not as good as before, and the surrogate threshold is 0.56. Again, do we use the surrogate PFS to predict a treatment effect on OS? Well, we can. We had, as I said, 12 trials that were independent from the trials we used for the calculations. And in these 12 trials, it turns out that we could not have predicted an OS benefit in any of them because none of them had a PFS hazard ratio less than the surrogate threshold effect. Remember, the surrogate threshold effect is 0.56, and the trial that has the lowest effect on PFS has a PFS hazard ratio of 0.63, which is not less than the surrogate threshold effect. Turns out that this trial actually had a survival benefit, but we could not have predicted that. Likewise, this trial here had a survival benefit. We could not have predicted that. And this trial here as well. And we could not have predicted that. So PFS is very poorly predictive of an OS benefit. You would have to have a major effect on PFS before you could be sure that you can predict a treatment effect on OS. And so again, this is shown on this slide. The prediction limits here are so wide that you just can't predict reliably a treatment effect on OS. And so this shows that in advanced disease, contrary to what I said before, and contrary perhaps to our expectations, PFS is not a very good surrogate for OS. And now we'll turn back to Everardo to look at some other situations besides the one that I've just shown here.
Thank you, Mark. So, my goal here in these last few slides is to discuss some of the disease settings for which research has been published, in addition to gastric cancer, which Mark has already discussed. So, in terms of the disease, the cancer types, there has been research using individual patient data in advanced breast cancer treated with taxanes and versus anthracyclines. So this is a specific set of 11 trials. Then for colorectal cancer, there has been research in the 5FU era, so to speak, and some subsequent research which I will also show you in a few slides. Then for advanced lung cancer chemotherapy trials, 7 of them. For ovarian cancer, again, chemotherapy trials, 4 of them. And then less evidence for hormone refractory prostate cancer in hormone trials from many years ago. So all of these are published, and they are all individual patient data meta-analysis. Now, what has been found is that there is a difference between these settings. For example, in advanced breast cancer treated with anthracyclines and taxanes, PFS is not a good surrogate for OS. even at the patient level, there is low correlation, but more importantly, at the trial level, the correlation is very low. So in breast cancer, it's fair to say, at least with these treatment types, that PFS is not a valid surrogate for OS using commonly used criteria. One thing we did not mention is that there are no universally accepted thresholds for considering whether a surrogate is valid or not. But of course, the closer to 1, the R square, the better the correlation. And typically, people consider values above 0.7, 0.75, 0.8 as indicative of good correlation. Now, in advanced colorectal cancer, there is much better prediction for PFS with regards to OS, both at the patient and trial level, and then not so good in lung cancer. Again, a bit smaller than with colorectal cancer. Good in ovarian cancer, despite the relatively small number of trials, and definitely not good in prostate cancer. In this analysis, PFS was measured not really as objective tumor progression, but rather PSA progression. So it's a different kind of endpoint, if you will. Now, Mark has shown that surrogacy has to be analyzed at 2 levels. So the individual level, or if you will, patient level surrogacy, establishes that the 2 endpoints are correlated, the surrogate and the true endpoint. And this is very useful for patient management. For example, if you have someone with early breast cancer treated neoadjuvantly and they have a complete pathologic response, this is a very good prognostic marker for this patient. Now, the trial level surrogacy establishes that the treatment effects on the surrogate and on the true endpoints are correlated. And this is what's more important to validate a surrogate, and this is what's useful to assess new treatments, as Mark has discussed. These 2 levels of correlation, patient level and trial level, at least for normally distributed data, are mathematically independent, which is not very intuitive. That's one of the reasons why there is need for evidence at both levels of correlation, patient level and trial level. Now, to finalize, I just wanted to discuss some issues about the evolution of surrogacy within a given setting, and then with different treatment types in the same cancer type. Now, the things can change over time. The associations between endpoints can change because of changes in therapy, evolution of cancer therapy. And colorectal cancer is a very good example. In the 90s, this tumor was treated with 5-FU or variations of 5-FU because they were the only sufficiently active agent. Then in the late 90s, irinotecan and oxaliplatin were introduced, and they really changed the outlook for patients. And then, of course, later on, the monoclonal antibodies came along, and since 2004, at least, bevacizumab and cetuximab have been available. So Mark and his co-workers did research that was published in 2000, and they described results in the 5-FU era. So they looked at several randomized trials and showed that they looked at response rate as a potential surrogate for OS in advanced colorectal cancer. And they showed at the patient level that there was a fairly good, as expected, a fairly good prognostic role for response, in the sense that patients that have CR fare better than patients with PR, and then stable disease and progressive disease, as expected and as observed in many tumor types. Now, at the trial level, there was not a very high correlation between the treatment effect on response and the treatment effect on OS. And the trial level R-square was only 0.38. Like I just said, there are no universally accepted thresholds, but this is certainly a low correlation between the treatment effects on these two endpoints. Now, more recently, there has been research that's still unpublished by the ARCAD group, in which we showed that with newer agents, most of the times 5-FU is combined with a newer chemotherapy, or in some cases with a monoclonal antibody. There continues to be a good correlation between responses and survival, if you will, at the patient level, but the trial level correlation is still low, and arguably even lower. Of course, there is an overlap between these the confidence limits for these two estimates, but it's true that there is no good correlation between the treatment effect on response rates and the treatment effect on survival. So in other words, response rate is not a good surrogate for OS in advanced colorectal cancer. Now, how about PFS? This is one of the diseases that Mark did not discuss, but our group did research on trials with 5-FU, irinotecan, and oxaliplatin. These were newer trials in comparison with the trials that I showed in the previous slide. And here there was a very good correlation between treatment effects on PFS and on OS. We're not showing patient-level correlations here, just trial-level correlations, and this is fairly good if we remember that in breast cancer the R-squares were much lower. So in this trial there are two estimates, because this estimate, which is fantastic, took into account one study that was somewhat of an outlier, and if this study was excluded, the estimate was lower. But still, there seems to be a fairly good correlation between treatment effects. Now, with newer agents, the same group repeated with the help with ARCAD data repeated an analysis more recently, and although the correlation is still there, it seems to have been attenuated. So one of the hypotheses here is that with the prolongation of post-progression survival, the association between treatment effects on PFS and on OS is being blunted, if you will. So this is a hypothesis, of course, but it suggests that that the correlation is less than it used to be, and it's reason to think that surrogacy is something that evolves over time, even if we consider the same disease setting, in this case advanced colorectal cancer. And finally, I wanted to mention something about different treatment classes, correlations that may be observed according to the treatment class within the same disease type. For example, in the analysis that I mentioned, we looked at treatment trial-level correlations between PFS, treatment effect on PFS and OS for three sets of trials. The ones indicated in blue are chemotherapy, red are anti-angiogenics, typically it's bevacizumab, and green is anti-EGFR, such as cetuximab and panitumumab. So if we look at all the trials together, they are all forming this cloud, and the correlation is obviously not tremendous, but it's really impressive when we break up the three groups of trials, because the correlations go in different directions. So this is still preliminary and unpublished, but again, it suggests that there are differences within a certain disease setting if we compare different interventions, and this is something that needs more research to be confirmed, but it's certainly something to be taken into account when thinking about surrogacy. And in breast cancer, the same thing. Comparing the study that I mentioned before for the association between treatment effect on PFS and OS for chemotherapy. At the time, there was a coefficient of 0.46. This is not R squared. This is rho, and here it's R squared. And here this is the comparison of trials that had an anti-HER2 agent added to the regimen, because these were patients with HER2 positive disease. So these are two different situations. This is breast cancer treated with chemotherapy, and this is specifically HER2 positive breast cancer treated with anti-HER2 agents. The coefficients here cannot be directly compared, but it seems that even for a highly targeted agent in breast cancer, the correlation between treatment effects is not good for PFS versus OS. So this is the final slide, and we hope to have some time for questions. So the conclusions we make is that there is a need for validation of surrogates, and there seems to be a growing need. If anything, it seems to be growing. Like Mark said, validation is best done using individual patient data because of the correlation between treatment effects that needs to be assessed, and because if one does that, it's possible to standardize the definition of endpoints, which is also a plus in comparison with published data. Then it's clear that a limited number of studies have been conducted thus far, and more studies are needed. For reasons that we still do not understand, DFS performs better than PFS when they have been both looked at in the same cancer type. And like I said in the final slides, surrogacy is context dependent, and it may change as treatments evolve, which is another reason to think about surrogacy research and continue to do it. So thank you very much. I will hand over to Vicky for a Q&A session.
Well, thank you very much for that insightful presentation. And now I would like to invite our audience to continue sending in their questions or comments right now using the questions window for the Q&A portion of the webinar. We have received some questions, so I'll go ahead and get started with those. And the first question we have here is for Dr. Boyes. Can the meta-analytical approach to surrogate validation be used even if only one trial is available?
Okay, well, very good question, thanks. Indeed it can. If you have a sufficiently large trial, the trial can often be broken down in smaller units, for example, by center or by country or by some other reasonable way of grouping sites. And in that case, you can look at the treatment observed in each of these units, whether they be centers or countries, and use the exact same approach as what we showed at the trial level.
Perfect, thank you for that. And our next question here is for Dr. Saad, and the question is, why aren't there more studies on validation of surrogate endpoints?
Well, again, a good question. We could ask ourselves the same. It seems that one of the main reasons is because of the difficulty in getting individual patient data from several trials. I have personal experience in working with Mark. We have tried to do that in certain settings for which this was not possible, just because we didn't get enough data, and we hope that data sharing will, in part, help expedite surrogacy research, because more data sets will be available.
Well, thank you very much for those answers. Unfortunately, we have reached the end of the Q&A portion of this webinar, and if you do have any further questions, please direct them to the email address showing on your screen, or if you would like a copy of the slides, please email Dominique at dominique.grizzard@iddi.com. Additionally, if your question was not answered during the webinar, IDDI will follow up. And thank you everyone for participating in today's conference. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up shortly on your screen, and your participation is greatly appreciated, as it will help us to improve our future webinars. Now please join us in thanking our speakers, Dr. Mark Boyes and Dr. Everardo Saad, and you can do so by clicking the link in the chat box. We hope you have found this conference informative. Have a great day, everyone.